Where are we and where are the dead animals?
So the dead animals we work on in this facility are all in little tubes and in freezers.
Hi Jay, thanks for taking our time to be here today. I'm wondering, what do you do at the LKCNHM Natural History Museum?
I am the molecular lab manager for the museum. I'm an entomologist at heart though, so I actually work on insects. But I also use molecular techniques to investigate biodiversity around the region and across the globe, and look at biodiversity patterns and other questions that you want to answer, which we can talk more about later.
Oh my gosh.
Hi.
It's hi to see you again. Nice to see you.
Yeah, you look like a familiar face. What are you up to?
Well, today I'm actually checking on the Cryogenic collection. Yep, so this is where I'm stored in materials. So it really cools up. Literally cools up.
So what is inside? It's like inside there you have animal tissue stored.
Animal tissues, blood, like the DNA, sometimes small specimens as well.
My first question is, what's the importance of the cryocollection?
So basically, we are actually storing all the genetic information of all your biodiversity, right? So this is because there's an increase in regards to genetic and genomic studies in the biodiversity.
We already know understand by now, the importance of having a natural history collection, because it allows us to look into the past to compare current diversity, right? The cryo collection, which stores all these biological samples that Marshall mentioned, is preserving the genetic matter of this biodiversity for researchers to use it for generations to come. So it's like longevity of like the information. Exactly.
And this kind of information are actually very important, right? So researchers can actually look into, like, say, the evolutionary history, right, the interaction between different animals in the food web, and including the environment, as well as looking at how these animals will actually respond to, say, changes, like, say, a climate change.
One more thing about having a cryo collection like this that's beneficial for not just Singapore but the region as well, is that, as we know, biodiversity loss is happening rapidly, right? And so to collect fresh material in order to do genetic studies for many species is not even an option anymore. If there are endangered species, for example, or they are in habitats that are hard to reach, don't want to have to duplicate the work of having to go out and do the collection of the specimens. If we can set aside DNA extracts or tissue from these samples that come in for other researchers to use.
What does it take to keep a cryo collection alive? Sweat, blood, and tears, babe.
And no, literally you had to stay cool all the time.
So if there is a power outage, these bags can actually maintain the temperature at a negative 170 degrees Celsius for 2 weeks if you don't open them. Also, did you get the Humpty and Dumpty names?
Oh yeah.
You both are entomologists, right? Specifically, so insects are known to be the most diverse of animals in the world, and there are lots that we haven't discovered. So molecular methods can help us expedite this biodiversity discovery process. Yeah, it also helps us narrow down a species ID, for example, that for groups that we may not have the taxonomic expertise to identify. There are also many other things that we can do with genetic data, but one of the biggest ways that having molecular samples helps us is that we can use molecular techniques to give us a working hypothesis as to what that species could possibly be. I'll take another. Handling tissue right now. So yes, this is where the magic happens. But before we can start investigating the genetics of these creatures that we work on, we need to extract the DNA. So what I've done is already done the lysis step, which is when we
use enzymes to break down the cell wall and other cell components so that we can free up the DNA that's caught inside. And then now we're going to use our DNA extraction machine to go through the steps that will help purify the DNA so that we have a final DNA extraction that we can use for our downstream work. So now I'm just setting up the system. I'm really glad we have this because it allows our researchers to free up the time that would usually take doing the bench work. To have it automated, it frees up like 1 hour of your bench time. And that in that 1 hour, we can do a lot.
Did you always know like this is what you wanted to do, or
It is a good question, no?
I always wanted to
Work on the environment. Actually, it's the only subject I got A for by A-levels.
I thought it'd be very sad if I didn't ever get to do bio again. Yeah, so I'll apply for life sciences here. The company is actually Oxford Nanopore Technologies. And the MinION is one of the sequences that they make that we use for doing DNA sequencing in-house. It is not the only sequencing platform. We also do engage third-party vendors to do some of the sequencing work. It really depends on the research question and the budget, the sequencing depth capacity that we need. They make it fancy.
Fancy!
So what molecular techniques can help with is to give you a working hypothesis to delimit your species. For example, we set up a Malaise trap and collect thousands of specimens. How do we then identify all of this? So one of the techniques we use is this large-scale DNA barcoding pipeline, where we use a particular gene in the organism's DNA to help delimit a species. So this particular gene, the CO1 gene, is what's mostly used for the animals we work on, but again, it can vary for different taxonomic groups. For example, plants, we use other genes. So this gene is known to be highly conserved within species and quite different between species because it's a gene involved in the metabolic pathway. So it's conserved within the species. It's part of the respiratory cycle, right? So if it's part of the metabolic pathway, it needs to be conserved in order for the organism that line to survive. The threshold for that really is dependent on the taxonomic group, but it then allows us to use this gene to then see if it matches with other known species in a reference database, something like GenBank, where there are lots of DNA sequences that we can then bioinformatically match to and possibly get a species ID if we're lucky. But most organisms are not sequenced and represented in the reference database. So that's one of the limitations of this technique, is that if you don't have a good reference database, it's not really giving you a species ID. But, it can help narrow down to maybe what family it could be in, what genus maybe it could be in, and that gives you a working hypothesis to investigate further. So what species that is. And then we can work with the taxonomy experts and say, hey, we think this is a new species. Is it? And instead of giving them an insect soup of, thousands of specimens and say, can you tell me what's in there?
Yeah.
We give them groups of samples, samples that we say, hey, I think this is possibly the same species. Could you verify it for me?
Right, so it's like a two-step kind of.
Yes. Yeah. So molecular methods are not the only way you can do this. What I'm saying is it helps us in our work in a time where we need to speed up the taxonomic processes. And then by doing this work, when we know what species it is, and when we publish our work, we put it up on open
access resources like GenBank so that other researchers, not just in Singapore, from around the world, can use these sequences to help with their work. Because again, understanding morphology is also a skill that one learns over time. So we use different ways to find the answer, basically. Yeah, that's it.
Thank you so much, Jane, for the very enriching tour.
Most welcome. Always nice to have visitors here.
Yeah, I see that. It's really nice. It's like a counter.